Hello everyone. So I'm Tim from Nanjing. My topic today is about extracellular antibiotic resistant genes according to EARGS. So the title is EARGS in the environment about its identification, fit, and the risk mitigation. As we know, um, extracellular DNA is a kind of DNA formed in the environment that are very typical. So the property of extracellular DNA is about its persistence. It can persist a very long time in the environment. For example, like the extracellular DNA in blood can keep for 10 minutes to 24 hours, while for the fossil DNA it can persist in 300 to 800 million years. So they persist in the environment and retains biological functions in various environments. And ERGS is kind of extracellular DNA. It arises from the lysis of alive cells or the creation of cells. And after it has been released outside cells, it can attach the substance in the environment and form ERGs with different physical status. And ERGs serves as critical reservoirs for bacterial resistance and playing a critical role in the spread of bacterial resistance. I focus on three subjects on ERGs. First is the extraction and identification. So there has been a few methods for eDNA extraction, like the CDAB-based method or magnet-based or column-based methods. These methods are commercial products that have been present, but they are not perfect. For example, there are not specific bonding for these materials with eDNA, and some hazardous agents are needed during the extraction. So there are new methods that are highly specific, environmentally friendly, and high yield are required. So here we establish a new method use the mesoporous magnetic beads-based approach. We establish the mesoporous magnetic beads with a diameter about 3 nanometers. So it is comparable to the size of DNA, and this led over a good specific of the materials to DNA, and we can see from the right panel that the materials has a very high recovery efficiency to DNA, and it also has a good specificity to DNA. But this measures is will decrease in specificity in very complex environments. So we further optimize the materials. Here we use the molecular imprinting. We know that the molecular imprinting has the property to recognize the target through a 3D dimensional cavities. So here we use the structure of DNA, the guanine, and we develop the guanine imprinted beads. So this bead has a very high recovery efficiency, higher than 90% without extra agents on DNA extraction. So it is higher than commercial products and the control. And we We also test the flexibility on various lengths of DNA, and it shows good to all kinds of lengths of DNA. And it also performs good in the presence of a coexisting substance like protein amino acid doses. And the mechanism is about this kind of a material has a stronger intermolecular forces with DNA compared to other substances. We use these materials to extract eDNA from various environments, and we found that the recovery rate is among the 70% to 105% for eDNA in rivers, plants, soils, and sludge. And we also found a very prevalent ERGs using this method is significantly higher than the commercial magnetic beam, and this made the ERGs is becoming predominant in mining environments. First, next, we use this method to explore the fate of ERGs in various environments. First, we extract ERGs in coastal crop soil, seawater, river water, and active sludge, and we found that the free present ERG and adsorbed form of ERG both present with a very high prevalence in the environment. And then we explore the generation of ERGs. Here we explore the process of wastewater treatment, and we found that in the influent, uh, the ERGs is relatively few, but with the process of treatment process, it becomes more and more, and in the final effluent, it becomes dominant, and the trends kept in the receiving river. So ERGs is dominant in the effluent and the river. And we further focus on the process of disinfection to on the effect of generation of ERGs. And we found that both chlorination and UV disinfection can lead to the production of ERGs, but they are different for the production. Like the chlorination will lead to the predomination of adsorbed form of ERGs, while this UV disinfection lead to the dominance of free formed ERGs. And then we explore the mechanism. We found that after chlorination, the cell fragments will be produced, and they can absorb the ERGs. So this made the generation of a soft form of ERGs. But for UV disinfection, the cell structure was basically intact after disinfection, and their adhesiveness didn't change significantly. So this resulted in a predominantly for type of ERGs, and these two kind of ERGs also lead to a different risk in the following environments. Generally, the adsorbed form of ERGs have high risk they can transfer through the horizontal gene transfer. And this kind of phenomenon also observed in exterior water bodies. And finally, we try to develop novel methods to distract and minimize the risk of ERGs. First, we should know that there is a lot of wastewater treatment process, but basically they are not efficient, including the biological process and inspection. They can't effectively eliminate ARGs, especially ERGs, in the environment. So the major problem for current process is they are not selective to ERGs because ERGs in water is very few, is little. So we need to develop a process that can selectively remove ERGs. So here we use the technique of molecular imprinting. We synthesize the uh, graphene like carbon nitride, we use it as a photocatalyst, and we coat the DNA guanine prints on its edge. So this made the material can select to bond to DNA, and then we give it is the UV light, and it can achieve selective gradation of ERGs in complex environments. So this is the characterization of these materials. And we can see that the materials performs good in simulated water with coexisting substance or realistic with water. But for the control, they decreased very significant. And because the presence of the molecular imprinting cavities, they lead to a very unique mechanism of destruction of ERGs. We can see that for the materials, the electrohole is becoming the predominant free radicals in the system, while for the control, it is the hydrogen. And this kind of unique mechanism make the destruction of ERGs more effective. We can see that the ERGs are destructed into short fragments. And further, we optimize the materials by using the CNT as the catalyst. And we further tested this material and found it performs good in environments, especially in very complex environments like the livestock wastewater. So, in conclusion, the ERGs include the boost-free and the soft form are commonly found in the environment. The current processes like chlorination promote the formation of ERGs and further facilitating the spread of ERGs. And selective degradation significantly reduces the potential risk of ERGs in complex environments. And that's all. Thank you very much.